Hi, everyone. I'm very grateful and honored to be here today. I would like to share with you about a method that our group recently developed, which we believe can revolutionize cyclic peptide discovery. So this method is called STREAM, which stands for Structural Ensemble Achieved by Molecular Dynamics and Machine Learning. So we will use molecular dynamic simulation results as our training data set. We will build machine learning models that can provide fast and high quality prediction of cyclic peptide structural ensembles. With these results, we believe we will be able to enable rational design of cyclic peptides with the structures that you want. We will also generate structures of cyclic peptides for virtual screening. And finally, we think this kind of structural information will help people distill experimental data to provide a predictive model and suggest next generation designs. So first, you're probably wondering, what are cyclic peptides? So shown here is a diagram that has the currently available drug modalities on the market. So on the bottom left is small molecules. So these are like aspirin or Benadryl. So these are small little organic molecules. They have very good pharmacokinetic properties, which means that you can just take the drugs by mouth. However, because these molecules are very tiny, they can target something small like enzyme binding pockets. But if you want them to interfere with, for example, protein-protein interactions or glue to a large protein surfaces, they are unable to do so. In this case, then people will look at something called biologics. So these are antibodies and other proteins. They are huge. They can target protein surfaces with high affinity and selectivity. However, because they are proteins in nature, you cannot just take them by mouth. They're going to just get digested by enzymes in our stomach. So you have to inject these molecules, which is very inconvenient for the patients. Also, these molecules are huge. They are unable to cross cell membranes, so they cannot reach intracellular targets. Then cyclic peptides are these things in the middle. So it turns out that there are fungi and bacteria that will make these cyclized peptide molecules. So people found out that, hey, these molecules, they can actually target large protein surfaces. They also can be orally bioavailable. So don't these molecules just combine the best attributes of the two worlds? So there are indeed some examples of cyclic peptides on the markets. However, there are only about 50 of them, and most of them are actually natural products or derivatives. So this concept seems to be very nice. However, it's very difficult to actually do rational design of cyclic peptide drugs. Why is it the case? So one of the challenges that we have identified is that it's actually very difficult to predict cyclic peptide structures. So for example, people know that, hey, I actually see this protein-protein interaction. I would like to interfere. So I know that this blue protein is using these 4 residues. They can actually glue to the binding to the gray surface. So these are called hotspot residues. Then if you want me to make a drug that can glue to the gray surface, great. I'm just going to have those 4 residues, right? And I can add some residue. I can have a cyclic peptide. But then the thing is, I need these 4 residues to orient exactly the way I see at the original protein-protein interface. If I actually know how sequence controls the structure of the cyclic peptide, then this problem is solved. Because I can say, hey, I designed this linker sequence. This is the cyclic peptide sequence. What does my peptide look like? Oh, it looks like this. Do these SESC residue, do these hotspot residue mimic what I want you to mimic? Oh, it does, and this will be a great candidate. Then if I design a cyclic peptide sequence poorly, I will still have those hotspot residues, but now they are pointing in some random directions. They are not effective at binding the target gray surface. Why are we not doing this? Scientists also found out that, hey, these are really funky molecules. When we throw them in solution, they adopt multiple conformations. So you will see many different structures. Each one has some kind of population. This means that even if you're willing to do experiments, even if you're willing to do solution AMR, for example, you cannot get an idea of what the peptide is doing because it's doing multiple things at the same time. As a computational chemist, there is a method called molecular dynamic simulation. I will run a simulation in my computer. So if this is the sequence number 1, I run the simulation, the peptide will change conformation as it would. And then by the end of the day, I look at my simulation. I will tell you, okay, 49% of the time I see the peptide does this. 36% of the time I see the peptide does this, and so on and so forth. So then you can once again do a structure-based design. You can compare, oh, this does not do what I want, does not do what I want. Oh, this does what I want. Only population of 2%. Change sequence, oh, now this population is 45%. So sequence 2 should be more promising than sequence 1. This method sounds so great, but no pharma company is using molecular dynamic simulation to do drug discovery. Why? Because this method is computationally very expensive. So with my computer resource at Tufts University, I can make prediction of 60 sequences. If I go to AstraZeneca or Novo Nordisk and say, hey, I can predict cyclic structure, and they say, oh, how many? I say 60 a week. They will say solid pass, right? So they want me to provide answers for hundreds of thousands of sequences in a day. Then this is where this new method comes in. So by playing smart, by running simulations and then analyzing the simulation results, we should be able to combine efficiency and accuracy. So what we do is you got to buy the bullet once. So we run simulations of some training sequences. Then we analyze the simulation results. Then we'll say, oh, this training sequence, I see these structures with this population. That sequence, I see this structure with these populations. Now, with all these results, we build a machine learning model to recapitulate what we've seen in the simulation. In the training sequence data set, in this figure, on the x-axis is the fitted population by the model. On the y-axis is what you actually see in the molecular dynamic simulation. Each dot is a specific cyclic peptide sequence adopting a specific structure. So you want y equals to x. So you can see that this correlation is really, really good. Now, how about if I give you a new sequence? So if I give you a new sequence, SBFAA, the model has never seen this before. In less than 1 second, the model will tell you that this is the top structure, population 19.8. This is the second most populated structure, population 15.8, so on and so forth. If you spend 1 day to run the simulation, you're going to get 19.2, 15.3, 8.5. So we test this method on 15 new random sequences. So you can still see that this y equals to x line is still trending very, very well. If you're willing to tolerate this kind of error, from now on, do not run simulation anymore. Just use the machine learning model. So we believe this novel technical solution can enable people to know the structural ensembles of cyclic peptides and use them to do structure-based design. We also believe that by combining the structural information with limited amount of experimental data, we will be able to build predictive models that can actually predict, for example, binding affinity, membrane permeability, and so on and so forth. Currently, there is a very, very, very strong interest in pharma companies in artificial intelligence. And also, even cyclic peptides or peptides can be considered a niche area. It's under constant, steady growth. There are, of course, people who are very interested in helping people develop cyclic peptides. So for example, Schrödinger, CCG, OpenEye, they all have computational suite that you can use for rational design. And what we found is that, well, they seem to work reasonably well for small molecules, but they do not provide high quality solution structure ensembles of cyclic peptides. For example, we have heard people say that, hey, they've been performing conformational sampling using some software. However, if they start with 2 different structures, the computer program gives them different results. So currently, the methods might be working well for small molecules, but they are unsuitable for peptides. I think they have very high efficiency, but the accuracy is not there. If you want to have high accuracy, you will go for molecular dynamic simulation that is so slow. By combining simulation results with machine learning, our method can achieve both efficiency and accuracy. So currently we have a patent pending and we're continuing working on more research and publications. We also participated in the I-Core Spark program at MIT in summer 2021 and the Bench to Bedside program at Boston University in fall 2021. And we have interviewed people in biotech and pharma companies and to collect their feedback. So it is generally understood that the tools are not good for cyclic peptides as they are with small molecules. So more needs to be done. And we believe that our method can help with better drug development through high quality structural information. We also currently have a collaboration agreement being finalized with a pharma company who I shall not name due to non-disclosure agreement. We are continuing to optimize our algorithm, and we want to be able to demonstrate the ability of using this method to discover binders for medically relevant targets. We are also very interested to demonstrate that this method, in conjunction with limited experimental data, we can predict, for example, membrane permeability for cyclic peptides. At this moment, I'm seeking help to establish partnership with biotech and pharma companies, and I will also like to have advice and help on recruiting executives and identifying and selecting appropriate business model. So thank you so much. I'm happy to answer any questions.